Welcome everybody to the last of the Kennisclips recorded by me. My name is Sarah Pringle. Today we're going to talk about the process of a tooth erupting into the mouth. Might seem fairly simple, but actually it's very complex because you need to get this hard structure, which is represented here. Here is the enamel, here is the dentin, through into the mouth. And of course, in between the teeth is not just a white empty space like here, but there is bone. You can see this bone on the right hand side represented by this yellow colour and the tooth here, and it is very obvious if you look at this size of this tooth that it will not fit through the space it needs to go through. This is called the gubernacular cord. What we need to do is to change this formation of the bone to a shape which allows the tooth, the space to move through the bone into the oral cavity. This is called the gubernacular cord. It needs to be made wider. You can also see it in preparations of the mouth. So here there are teeth which have obviously already erupted, but these are the gubernacular canals for a second set of teeth to come through. What I'd also like you to think about, and which actually we don't really know the answer for very well, is even when we have managed to remove this bone, what is the force which actually moves the tooth up into the mouth? Is this just because of the growth of the tooth? Is it the force of the root pushing it up? Is there actually something to do with the bone being made wider that releases some pressure? We don't actually know which force it is which moves the teeth into the mouth. For example, in mice, which were engineered to not have any roots, the teeth still come through into the mouth. So it cannot be just one factor. This is a fairly complicated process, as you maybe can imagine, because we need to dissolve away some hard bone tissue. I'll take you through it slowly. So starting at the bottom, these are all figures adapted from the Berkowitz textbook. This process starts in the stellate reticulum on the right hand side here, which should be a name which is still familiar from the types of cells hanging around in the development of teeth. In the stellate reticulum, a variety of growth factors are produced, including PTHrP, IL-1 alpha, and TGF-beta 1. This is parathyroid hormone receptor protein, something like that. These signals produced by the stellate reticulum signal into the blood for the recruitment of a particular type of cell from the blood. So this is important. These cells that we need in order to chew away some bone and make that opening bigger, the cells that we need are called osteoclasts, but they are not readily made. First we need to make them. First we need to recruit progenitors from osteoclasts into the right part of the developing tooth where the bone is to be removed. So these signals from the stellate reticulum recruit, so attract in precursors for osteoclasts. So precursors, progenitors, so think about the cells that are capable of turning into osteoclasts. And there's a whole signaling network of things which induce this process. So mononuclear, one nucleus here. Osteoclast precursors are attracted by the stellate reticulum, so the factors that were made here, and also factors that are now made by the dental follicle. We're now moving into the dental follicle. This is happening in the dental follicle. Are attracted from the blood into the dental follicle. So now you have a different panel of cytokines and chemokines and growth factors and all kinds of things which work together to recruit these cells into the dental follicle and also to activate them, because this cell here will not remove any bone. What happens and what needs to happen is the mononuclear cell fusion. So now you can see we're in the dental follicle. This whole process here has been happening in the dental follicle, and we see fusion of these pre-fusion types of osteoclast precursors. So between here to here, because of these signaling pathways, the cells become a little bit more mature. They start to express different receptors on the outside. For example, the rank receptor, an OPG receptor, which is really important for the maturation of osteoclasts. When we merge, in this case, a few of these cells together, let's say 1, 2, 3, 4, 5, we generate what is called a multinucleated cell. So a multinucleated osteoclast, there are more than one nucleus, expressing receptors associated with functionally mature osteoclasts. These cells are the cells now capable of dissolving away bone, which is what we need to do in order to make the gubernacular canal bigger to let the tooth through. So number 3 on the right hand side, fusion of mononuclear cells into osteoclasts stimulated by rank L, so I'll come back to this, produced by the alveoli of bone. So the bone is what we need to get rid of. Rank L, rank is the receptor which I was talking about on the outside of the osteoclasts. Rank L is the ligand for the receptor. So the generation of these functional osteoclasts is stimulated by the alveolar bone itself, by the production of a ligand which activates these osteoclasts into their final functional form, where they're capable of chewing away bone. These cells look pretty weird. They look a bit like a spaceship or some kind of cell that you probably wouldn't think is a very healthy cell, but they are, and they're also really beautiful to see in real life in microscopy. So let's have a look at what they look like. So again, the title, osteoclast activation by osteoblasts. Osteoblasts are the cells which make bone. Osteoclasts for chewing, are cells that digest bone. So if you try and picture this sequence of events back into the slide before, here is rank L, which I was saying was made by alveolar bone. There's all this structure around the bottom, binds to the rank receptor on the outside of the pre-fusion osteoclasts, stimulates the fusion into multinucleated osteoclasts which are mature and can then kind of sit into the bone like you see here. So they have all these, they form like a seal, which is really important to their function, because the dissolving of bone, you need to use some really acidic substances to dissolve the bone, and the bone directly underneath this sealed capsule is the piece which will be dissolved. In terms of microscopy, you can see them very easily. So this is bone, obviously. Hopefully you're a bit used to looking at bone. And these guys are all activated multinucleated osteoclasts. So you can see 1, 2, 3, 4, 5, 6 different nuclei, and that kind of a like a hole or a pit of bone where the osteoclasts are sitting in and chewing away. So it's by this process which the alveolar bone, which is too narrow for the teeth to erupt through, is chewed away to allow space for the teeth to emerge and to erupt into the oral cavity. This process begins with signaling from the stellate reticulum and then involves also signals from the dental follicle, where the precursor cells are recruited to, and is eventually ended by the attraction of the pre-fusion osteoclasts towards the bone through rank and rank L signaling. What we want to talk to you now about in the second little part of this Kennisclips is the gingival sulcus. So if you're looking at this diagram here, for example, of a tooth erupting through these stages from A to G, you can see that the oral epithelium merges with the reduced enamel epithelium to form a continuous barrier between the oral cavity and the teeth around the outside. This is really important for you as dentists, because this is the cell layer which forms a barrier between the outside world and bacteria and infection and the blood and the inside of the body. So you'll become very used to checking the health of these areas of the teeth. This is called the sulcus, the gingival sulcus. Sulcus basically just means a gap in the tissue, and it can become very inflamed and give problems and access for bacteria to the blood of the patient. So we want to spend just one more slide looking at how this is built up and what is important about it. Epithelial fusion, the title of this slide, leads to gingival sulcus formation. What do we mean by this? So, like I just said, a sulcus is a groove or a space or a crevice. So let's zoom in on this area here. Again, this will look familiar now, dentine, enamel. All of this together, the abbreviations are oral epithelium, this is the sulcular epithelium, this is the junctional epithelium. So there are different types of epithelial cells which you can recognize based on their morphology, and together form the gingiva. The whole thing together is the gingiva. In a healthy situation, you don't see very much fluid flowing from the gingival into the sulcus, so into this space here. This would be the gingival sulcus, the space. It's a small space and it's not full of fluid. In a more diseased situation, there will be more fluid flow and inflammation, immune cells into this area here, leading to potential damage of the teeth and also further into the tooth structure itself. But okay, let's go back here. So the sulcular epithelium, this layer of epithelial cells here, which you can see on the right hand side is here. So this is this would be dentine. Enamel should be here, but because it's such a hard tissue, it's been removed for the processing of the tooth. So this is not one huge sulcus. This is a gap where the enamel should have been. This is a healthy situation. Here is the oral epithelium, you can see all the striations, and here is the junctional epithelium. So you have these two types of epithelium are different to each other. They might look fairly similar if you go with your eye from the bottom to the top, junctional epithelium, sulcular epithelium. Okay, how do I know what the difference is here? But the difference is the attachment to enamel or not. So the sulcular epithelium is not attached to the tooth. And this is a permeable type of epithelium which will allow fluid and immune cells and cytokines into the connective tissue here, further kind of encouraging an immune or inflammation response in a non-healthy situation. The junctional epithelium, JE here, forms like a collar surrounding the tooth and extends from the cement enamel junction, CEJ here. So this is the joining between this is the cement, like we talked about in root development. This would be then the primary cement, if you remember correctly, around the outside of the tooth, and the enamel. So the CEJ is obviously the gap between the two, or not the gap, the join between the two. The junctional epithelium bridges this gap, this change in tissue type, and extends from the bottom of the gingival sulcus upwards, forming a hard attachment to the tooth. So to summarize, we have talked today about two main features to do with teeth eruption, and that is the resorption of bone by osteoclasts, activated osteoclasts, which are recruited to the teeth zone, to the bone, as precursors from the blood by way of different soluble mediators, including rank and rank ligand, and also about the gingival sulcus, which is this layer of epithelium between the gingiva and the enamel on the tooth, which will be important clinically for you as future dentists as a site of very common infections and inflammation.